Alrighty, we are going to, since we have done replication at length, it is now time for us to figure out what it is that this DNA actually does. We know how it copies itself so that we can send copies of this genetic information to offspring cells, okay? But we have not really discussed how it is that this genetic material actually creates a living thing and allows that living thing to function on a day-to-day basis. So that's the job of this part of our study. So that said, let's start this little set of thoughts with the idea of going from a gene, which is DNA, to proteins, which is what DNA is the instructions for making. Okay? Genes are the instructions for making proteins. And essentially, proteins are you. So DNA is the instructions for making you because it's the instructions for making all of the proteins that make up you. Okay? Now, so this then is all about how genes work, how they actually serve as those instructions to create those proteins. So, just what we just talked about. DNA is the directions for building proteins. And then those proteins do all the work of creating our cells and ultimately our entire bodies. And this is true for all life on Earth, whether you are a teeny tiny bacterium or a great big elephant or a great big redwood tree. Those proteins create our cells, which then create us. Now, what we are going to be focusing on is this, going from DNA to protein. We're not going to worry about this stuff out here. We're worrying about going from DNA to protein. This is called protein synthesis. It is also called gene expression. I have expression of genes here, but gene expression meaning what happens when those genes express themselves. What do they create? What protein do they make when a gene expresses itself? All right, so we know how to get from DNA to DNA, okay? That's the idea behind replication, okay? And we've gone through that and all the enzymes involved in that, okay? And please remember this replication is done in order to pass DNA to offspring, but nothing is really done with the DNA. It's just passing it on to offspring. How is it that we get from DNA to actually protein? Well, RNA is the answer to that. RNA is the molecule that is going to be our transition to bridge us over from the DNA that is protected in the nucleus to proteins, which are created in the cytoplasm and may either stay in the cytoplasm of a cell and work in there or be secreted from that cytoplasm and used to do or create other things, okay? At any rate, it is proteins that actually give us our traits, okay? Certainly the DNA is the instructions for making the protein, but what we see when we look in the mirror and we see our brown hair or our blue eyes or whatever, we are seeing the traits that those proteins give us. Okay, so transcription right here is the process where we go from DNA to this in-between molecule, this RNA. So that's what we're going to focus on first. That's the first hunk, and that's probably all we'll go through in this particular PowerPoint. The second half of this step is translation. We're going to focus on that. There we go. Translation is going to go from RNA to protein. That's the second half of this. Okay? Altogether, you probably should get this in your head, that this whole concept going from DNA to an RNA molecule to an actual protein is called the central dogma. And, once this was figured out, that was a pretty big deal, that genes code for proteins. And so you should keep that in mind. I'm going to see if we can come up with another way to go through this rather than dealing with this in a PowerPoint. So let me skip over this real quick. Skipping. Beadle and Tatum, which is whose work I just completely skipped over, two scientists who originally came up with the idea that their original idea was genes code for enzymes. Each gene segment of DNA, if you will, codes for a particular enzyme. And these guys came up with that. So much as Watson and Crick came up with the structure of DNA, these guys were the ones that said, well, this is what DNA does that's really important. It gives the instructions for making our enzymes in our body, without which our bodies couldn't function. That has since been broadened to one gene actually codes for one protein. So remember, protein is a pretty large category. Protein is a really big category. And enzymes are just one type of proteins. There are lots and lots of others. So genes code for proteins in general, and this may change even further still as we learn more and more about genes and how they work. In fact, we are figuring now that one area on the genome, on the genetic code, can actually code for several different proteins, which is a little mind-boggling, but we'll talk about that. All right, now what we need to do is understand the first part of the central dogma. So we need to understand if DNA gives us RNA, which then gives us proteins, what we're going to do is understand this process here, which is transcription. Okay? Now, we are going from DNA language to RNA language. That is like, I don't know, maybe going from Richmond, Virginia to jolly old England, okay? We speak the same language. We've got a slightly different accent, but we could understand each other. We can understand people, even though they may speak with a British accent. We can understand what they're saying most of the time. That's how you need to think of DNA and RNA. It's the same basic language, a little different twist, okay? So RNA, that said, is very, very similar to DNA, except for in three small ways, okay? First of all, the sugar involved is not deoxyribose as it is in DNA. This is the sugar in DNA. It is just a simple ribose sugar in RNA. And that really has not got a whole lot of bearing for us, other than we can remember that's how RNA gets its name. This will be very important in how we work with this molecule. In RNA, there are no T's. There's no thymines. You know how A in DNA always pairs with T? Well, in RNA, that's not true. In RNA, A always pairs with U, which is uracil. There's no thymine. Okay, and up here you can see that this diagram is showing the bases are all the same except for that right there. In DNA, you've got your T's. Okay, and very importantly, RNA is a single-stranded molecule, as you can see clearly illustrated here. We've got the double helix DNA on the right and the single twisted strand of the RNA on the left. Okay. Also, there are several types of RNA that we can talk about. We will be talking about three types. mRNA, this is the one we'll spend most of our time talking about, but we will also talk about tRNA, and we will spend fairly little time talking about rRNA. rRNA is, R is short for ribosomal RNA, and this is RNA that actually goes into building up the little tiny ribosomes that you might remember. If you've got a cell and there's our nucleus, you've got this endoplasmic reticulum, and there are these little ribosomes that sit on it, and they're what makes the proteins. Our RNA is part of that. The other part of a ribosome is protein itself. So ribosomes are made of our RNA and protein, and that's probably about all we'll talk about them. Okay. Now, why have this intermediate? Why not just go straight from DNA to protein? Why have this RNA jazz in the middle? Well, it does provide some protection for the DNA molecule. DNA is the precious genetic information, and generally speaking, we don't send our most valuable documents just out into the world. If we need to send these, the Constitution, for example, out into the world, we make copies. We don't send the original out for everyone to peruse. It could get damaged. So instead of sending the DNA out into the cytoplasm of a cell, we keep it safe in the nucleus, and we send a kind of copy of it out into the cytoplasm. Also, we can make many of these RNA copies and send those out into the cytoplasm, and that can allow us to make a lot of product that we may need. So that's another reason. All right, so in the nucleus then, we're going to have our DNA, and we are going to make something called mRNA, and as you might remember, the M stands for messenger, because this is going to be acting as the messenger, taking the genetic message from this DNA molecule and sending it out into the cytoplasm, where it will be used to give the instructions for making the protein, which is ultimately what we want. This thing is the thing that actually does the work. Proteins are the things that do the work. This is just trying to get the instructions out so we can build that thing. Okay? Now, it is important to understand ribosomes are very important in this whole process. These are the little teeny tiny factories in cells. And bacteria have ribosomes, we have ribosomes. They're different a little bit in bacteria than they are from us, but nonetheless, they're still ribosomes. And those are the little protein factories. The message has to get into the ribosome, and protein comes out. Okay, so they are the protein factories, and they are going to make these strings of amino acids, which that's what that is supposed to be showing. All those little A's are standing for amino acids, and that's basically a protein. Okay, now, again, we're not quite ready to talk about this yet, but we will be. All right, transcription then. We are going to be making something that is rather similar to DNA, but not quite exactly the same. And the way we do that, it looks, initially at least, a lot like replication. Okay? We will open up the DNA double helix, and we are going to kind of do something, as you can see here. We're creating this mRNA strand by pairing up nucleotides with the existing DNA template. Now you can see here the A on the DNA, which would normally pair with the T, is when we make RNA going to pair with a U, but otherwise it's a really similar process. Okay, but only one of the two original DNA strands is transcribed, the side of the DNA that actually has the gene we want copied. Okay, so the untranscribed side of the DNA is called the coding strand, and the transcribed side is called the template strand. So in other words, this strand is the strand where the enzyme is actually saying, okay, here's A, I'm going to put a U next to that, here's a C, I'm going to put a G next to that, here's a G, I'm going to put a C next to that. But if you look over here, this side that's not doing anything, if you look, except for where this has a T, and it would, if this were RNA, it would be used. This is exactly what's being coded for down here. So this is the coding strand. This is really kind of, if you want to cheat, you can say, oh, well, this is exactly what my RNA is going to look like, except for it's got T's instead of U's. And this is the template strand, and we're going to make the complement of the template strand right there. Okay? All right. Now, remember we had something called a replication bubble? Well, now we're going to have a transcription bubble, okay? And that's where the making of the RNA takes place. And the enzyme this time that is in charge of everything is an enzyme called RNA polymerase. Remember, before we were dealing with some DNA polymerase and two of those and a helicase and a ligase, and we're not going to have all those players this time, just RNA polymerase, okay? But just like DNA polymerase, we are going to go in the 5' to 3' direction. In other words, we still have to hang on to a 3' end, okay? Now, there are 3 different RNA polymerases. RNA polymerase 1 only transcribes RNA that's going to go into making ribosomes, rRNA. RNA polymerase 2 only transcribes those genes that are going to go into making mRNA, which is the messenger RNA. RNA polymerase 3 only transcribes those genes that are going to make tRNA, and we'll talk about the importance of that shortly. So what we're going to spend time talking about most of the time, though, is this RNA polymerase 2. And what you see in this diagram is a picture of the RNA polymerase running along the double helix, and here comes RNA being squirted out the side, if you will. Now, this enzyme, this RNA polymerase, each of these enzymes has a specific, what is called a promoter sequence. What that means is a sequence on the DNA that it recognizes. So this is a sequence of nucleotides. DNA nucleotides, a particular sequence that the enzyme will recognize, and it knows, hey, I'm supposed to attach right here on the DNA molecule because I'm seeing AACGT or whatever the particular sequence is. If it sees it, it knows it's supposed to attach and start its transcribing. Okay? Alrighty. Now, if you'll recall, here is a chromosome, a single strand of DNA right here, okay, and we'll make it a double helix, sort of, have an imagination here, okay, and this is a really long molecule, and there's a whole lot of genetic information on that. Maybe from this point on the DNA strand to here codes for someone's eye color. Maybe from here to here determines their blood type. Maybe from here to here determines, oh golly, their height. And I'm oversimplifying this, and so on. So there could be several traits running at different positions on this chromosome, okay? So what determines where one of these genes starts and stops? What determines that? Okay, the promoter region is what we're looking for, and we already mentioned it in the previous slide. It is a sequence of nucleotides, AATTG, whatever it is, a particular sequence on the DNA that is found before the beginning of the gene that we want to transcribe. Okay? It is a little sequence of nucleotides that tells RNA polymerase, hey, stupid, attach here. This is where you need to start transcribing. Very often, it is referred to as the TATA box because, as you can see here, TATAAA is a common component of the promoter sequence. So it has been referred to as the TATA box. Do with that information whatever you will. Yes, it's the TATA box. Okay? So this is what tells the RNA polymerase molecule plus these things called transcription factors, which help the RNA polymerase bind. And if you'll recall, we talked about back when we talked about hormones, we talked about those lipid-based hormones that can get into a cell, like cholesterol-based hormones that can go into a cell and go into a nucleus. Well, this is part of what those hormones would do. They would attach to proteins called transcription factors, and that hormone would cause these to attach here, which causes RNA polymerase to attach here, and then this gene would become activated, and we'd start doing some transcription, and that gene would produce a product we need. So you should be aware of what transcription factors are. They help RNA polymerase attach at the promoter, so you should recognize that term, and it can't hurt to recognize the TATA box. How could you forget the TATA box? I don't know. All right, there are 3 stages of transcription. Getting started, called initiation, going along with it, which is elongation, and it looks a whole lot like replication, but it's not, and termination, ending it. Okay, so initiation means we have to bring all the things together that we need to get started. So that means the transcription factors that are going to help the RNA polymerase attach, and then the particular sequence on the DNA so everything knows where to attach. So the transcription factors, the RNA polymerase, and the promoter. Those are the 3 things we need to bring together, and this diagram is showing the little purple bubbles here are all the transcription factors required, and this big thing here is RNA polymerase, and then the promoter sequence would be a spot on the DNA right in here. Okay? So the transcription factors bind to the promoter and trigger RNA polymerase to bind. Now, once we've bound everything up, RNA polymerase is just going to start going on down the DNA and adding nucleotides that are complementary. Remember, U replaces T. And we're going to grow in the 3' direction. Okay? When we end, basically there will be a terminating sequence on the DNA. Now, this is kind of slightly wrong. It results in this on the RNA. Obviously, if it's on the DNA, then it would be the complement on the DNA, so this would be the complement on the DNA, but you will see this on the RNA. That's the terminating sequence, and that will tell RNA polymerase, hey, you've gotten to the end. So let's say the terminating sequence is somewhere right in here, and then so that would tell us to stop, and we don't need to transcribe any more of this DNA molecule. We've got the part we needed. It was right there. Okay? So once you get your completed RNA transcript, there it is, we made it, it's in red right here, it's not done yet. It might be done if it were in a prokaryote, if it were in a bacterium, but it's not done yet if it's in if it's in a eukaryote. We have to polish it up. We've got to finish it off. We've got to make it ready to do what it needs to do. And that's called RNA processing. This is kind of part 2 of the transcription process. Again, only in eukaryotes. The RNA, when it is first completed, is not usable. We've got to do some things to it. The first thing we have to do is modify the 2 ends of the molecule. So if this is my RNA molecule, okay, I got to do something to the starting end and the ending end. Okay? All right. Also, there are regions within this RNA transcript that evidently are not needed. Why are they there? Well, we'll talk about that. They use these pieces of RNA that are not supposed to be there, were once called junk. That's so typical human. We don't know what it's for, so therefore it must be junk. We know now that these stretches of RNA are probably not junk, but they are cut out, and the remaining DNA is going to be spliced together, and we're going to talk about that. So, eukaryotic genes have junk? Well, probably not, but when you get a piece of RNA, the parts that you keep and don't get cut out but are retained are called exons. Okay, so this is the real genetic information that we want for a particular trait, a particular protein. Maybe it's what's going to give you your eye color. This is called exons because these are the expressed DNA that are going to code for the trait. Introns are the stuff that's in between the coding parts, and this is the junk. It's probably not really junk, but that's what it got called. So it's the in-between sequence and what must get cut out because it's not part of the actual coding part of the DNA. So the introns come out. Okay? Now, when we remove the part that's junk, we've got to glue together the pieces that remain behind. And this cutting and gluing is called splicing. Okay? The introns are cut out and the exons are joined together. Okay? So if you look at this, this is the RNA molecule that is initially produced by the transcription process. And this part, called an intron, is not needed for this particular gene, nor is this part. So we would have particular enzymes that would come in and slice these parts out, and then these remaining exons here, here, and here would get attached back together again. Okay, so this is mRNA splicing. The primary transcript, so look, here's the DNA. Okay, this is the DNA we made the original mRNA from, but all we really want for this particular gene to do its work are the green parts. So we're going to cut out these orange parts, and we're just going to leave this. This is the processed, yeah, I can spell, no I can't, processed mRNA. This is the completed thing down here. This is mRNA, the primary transcript right there that has not been processed. It's also called pre-mRNA. After splicing, we've cut out the introns. This part is the mature or processed mRNA down here. Okay? So that's DNA, primary mRNA, or pre-mRNA, and that's the spliced mRNA. Okay? Okay. And if we had more time, we could talk about these guys, but we don't, so I'm skipping it. Now, yeah, not Smurfs. These are called SNRPs, okay? The things that do the splicing are actually RNA molecules. Now, isn't that funny? I thought proteins were enzymes. Well, let's talk about what these are. SNRPs are actually small pieces of RNA, okay, and proteins. So let's not get too carried away. It's RNA and protein complexes, okay? And they come together to make things called spliceosomes. It's like a little cutting and pasting machine, okay? So there are several SNRPs, if you will, which are small nuclear RNA or RNA molecules combined with proteins that recognize certain sequences that need to be cut. And so what they do is these, here's some SNRP, okay? These things come together. They bend the RNA transcript in such a way that it can be cut and then knitted back together. And this is the intron that we took out because we don't need it. And then the SNRPs are kind of go on their merry way again. So SNRPs make spliceosomes, and spliceosomes are the things that actually do this cutting for us. Okay, now what you need to remember, as we have talked about since very early in the year, proteins being the group of organic compounds that gives us enzymes, which are the things that do catalysis, that give us catalytic reactions. But in fact, it has been found that RNA can also act like an enzyme. It has catalytic properties, okay? They aren't protein enzymes, but they do have catalytic properties. The spliceosomes are a case in point. Now, the RNA in the SNRPs that we've talked about is thought to have catalytic properties, very much like enzymes. And these kinds of RNA molecules have come to be known as ribozymes, RNA molecules that act as enzymes. In some organisms, RNA splicing can occur with no protein whatsoever, because the intron can act, the junk DNA can act, junk RNA can actually cut itself out because it's functioning as its own enzyme to cut itself out. It's pretty staggering. The whole point of this discussion is that it's got really important implications for how RNA might have been really important early in the evolution of life. What is it? If DNA needs enzymes in order to copy itself and do all it needs to do, then how could life have evolved? Because you've got to have one thing before you can have the other. You have to have the enzymes to work with the DNA, but how could you get the enzymes without having the DNA? It's you got a chicken and egg thing. But what if there was a genetic material that could catalyze its own chemical reactions, its own cutting, its own pasting, a lot of its own activities? RNA fits that bill. What is it about RNA that allows it to act as a catalyst? Well, for one thing, it is single-stranded. DNA being double-stranded, its bases are very protected, and it doesn't mutate a lot. It's very stable. RNA being single-stranded means that the shape is going to be much more highly variable. One part of the molecule can actually base pair with a complementary region down the molecule. The molecule just bends and the two complementary parts could come together and it could take on a particular shape. Well, what do we know about enzymes? It's the shape that makes them work. And so it is thought very much that some of these bases have functional groups attached that could act as catalysts in the right conditions. So this sort of structure of RNA, this single-stranded thing that can take on a unique shape and even have functional groups that might do little activities, is really, really similar to protein. So it's kind of important to understand that RNA can have catalytic properties, and that is going to be important, it is thought, for the evolution of life on Earth. And in fact, it's thought that perhaps RNA was really the first genetic material, not DNA. So the idea that biological catalysts can only be proteins is in fact an obsolete one. All right, so here's another idea that's going to complicate things: alternative splicing. Depending on what you decide to cut out of a stretch of DNA, you can get an entirely different gene. Okay? If you look at this, for example, let's say we cut out, so here are our exons, the brightly colored things here. Okay? And we get, and this is the DNA, of course. This is the RNA. We copy that DNA to get our RNA, but depending on what we take out, if we take out this green, then we get this gene, but if we leave the green and take out this, we get that. All because we took out something different and left other things in. This might code for a completely different protein from this one, all because we tinkered with how we splice this thing together. So, the bottom line is more than one type of protein can be coded for by the same region of DNA. So, that question, when is an intron not an intron? When we decide it's going to be an exon instead. It's getting increasingly difficult to define exactly what a gene is. It does do us a big favor because it explains something that had puzzled some. It used to be when I first started teaching, we taught that, oh, there must be about 100,000 genes in the human genome. We now know maybe there's around 25,000. How could beings so complex have only about 25,000 genes? That's not a whole lot more than a fruit fly. Maybe less. I can't remember how much fruit flies have. So the only thing that really explains how we could be so complex with this few number of actual genes is that you can get lots of different proteins from the same gene depending on how you cut it and paste it together. Pretty complex, pretty crazy, pretty hard to figure out when you're trying to figure out what gene codes for what, and all of a sudden now you've got to figure out how to cut and splice, and so layers and layers of complexity as we learn more about genes. All right, more that needs to be done. After we splice, we also need to protect the tips of the mRNA from being degraded too quickly, so we're going to add stuff to the tip ends. If we don't, enzymes in the cytoplasm are going to degrade the mRNA. So we're going to put something called a 5' cap, and that's right here on one end, and something else called a poly-A tail, which is just a series of adenine ribonucleotides on the other end. Usually you can see right there, it could be up to 250 adenines long. Okay, so these caps are simply to protect the mRNA from being degraded. All right, so that is pretty much everything you need to know about transcription. And next we need to find out all you need to know about translation. So I think I'm just trying to, yep, that's where we're headed. So thanks for watching, and I'll see you in the next podcast.